So let me first tell you what educational neuroscience is. And it really grew out of the tradition of cognitive neuroscience of trying to figure out how the brain supports the mind, right? The search for correlations between mental processes and brain areas. But the twist was to focus on the kinds of cognitive skills that you need in the classroom. So specifically reading and math. Which parts of the brain do reading, which parts of the brain do math. And it's grown to think about how experiences that kids have in the classroom impact their brain and impact their mind. Okay? At first, these studies were correlational. Correlations between how kids are doing in school with their brain structure. I have one of these studies, and how this maps onto their cognitive skills. And I think where the field is going is looking at the deltas here. So when the classroom experience changes, we have new educational interventions, how does that actually change the brain and change the mind? So what I want to do today is similar to Joe. I want to dive into a couple topics pretty deeply, and then I want to broaden out to sort of the more translational issues that the field is facing. The first thing I'll talk about is a current controversy about how exactly the brain can change with intervention. Then I want to tell you some new findings from our lab to give you sort of a template study of where the field is going. That's really self-congratulatory. I don't mean it that way, but I think it brings up important issues. And finally, I want to tell you sort of where I think the field is going, both as a basic science and as a translational science. And I will say that a lot of these issues are not specific to education, and I invite you to sort of draw parallels and similarities between education and law and some of the other fields that you have expertise in. So let's start with controversy. Always a good place to start. And the controversy is this. So when kids with learning disabilities, they get educational interventions, how does that change the brain? Does it make their brains more like typically developing kids? Or do they show changes that are compensatory, completely different from typically developing kids? Right? This is not a small issue because it's really core to what the field is trying to do of predicting which interventions will work, right? And assigning interventions based on a kid's brain patterns. Right? So if sort of the gold standard of education working is around normalization, right, we could be wrong. We could actually need to know that what kids are doing when they're getting better is about compensation. So to make this more concrete, let me tell you a story about reading. So those early studies I was mentioning where we figured out what the brain is doing when it's reading mapped a circuit that brought together visual areas with areas for language comprehension and language production. Right? And those areas are brought together structurally through big white matter tracts in the brain. This yellow one in particular is the superior longitudinal fasciculus, sometimes called the arcuate fasciculus. Those differences are minor, so I'm just going to call it the SLF to make things easy for myself. So the SLF is connecting all of these language areas. And because language in a lot of people, most people, is left lateralized, there's been a lot of focus on the left hemisphere. So what we saw a few years ago now is that before kids even start to read, there's a correlation between their phonemic awareness, their ability to hear sounds, and the structure of the left SLF, that connection between these language areas. The kids with better ability to blend words together, like k, t, the kids with the better abilities to blend those words together had a better organized left SLF, a better organized white matter tract. This is before they start to learn how to read, so this is not an effect of these kids reading less because they hate it, right? This is really something that looks like it's very, very early. What's also very interesting is that if you now take kids who have dyslexia and you look at who are the kids who over the course of an academic year really learn well, right? The prediction comes not from that left SLF that looks like very early on there's something that's a little bit different about it, but from the right side of the brain, right? Suggesting that actually those compensatory mechanisms are explaining these individual differences and whether kids are then successful in learning how to read. The study that came out last year that I want to tell you about tests this very directly. And so they take kids and they follow them through an 8-week intervention. They scan them 4 times at baseline in session 1, at 2.5 weeks in session 2, 5 weeks, session 3, and at the end in session 4. And they're tracking these changes in white matter tracts. I'll show you data specifically for that left SLF, that left language tract, but this is true through several tracts of the brain, where they show changes. And those changes, you can see from the dark green line to all of the other lines, happen in the first 2.5 weeks. These brain changes are very, very fast, and they hold across the entire course of the intervention. And then we can ask, do these changes make these kids look more like typically developing kids or less like them? And the answer is less. So they start off here, okay, and over time, their reading scores go up. That's good, intervention is working. But the mean diffusivity, this measure of the white matter tract, is going down, right? Down and not up, not to look more like typical readers. So their brain is doing something different, more like compensation, less like normalization, right? So if we said, okay, effective interventions are going to make kids look more like this, right? That would be the wrong benchmark, the wrong goal, based on this small study. I will say that this controversy is far from solved. A study in the math domain from a few years ago now showed precisely the opposite, which is true in many neuroscience fields, where they looked at actual functional activation during a math task in kids with dyscalculia, a math learning disability. And what they found pre-intervention is that there were major differences between the learning disability group and the typically developing group, and by the end they were gone. So what might be happening is that intervention leads to structural rewiring, which actually leads to functional normalization. These two are not mutually exclusive. What could also be true is that one of the studies is wrong, right? And what I would hope is that this is such an important question that we start to see much bigger studies. That reading study that came out last year had 24 people in it. This study had 15, right? So we're staking a lot on these really small studies. But what I would say is, even if the precise pattern is still open to investigation in larger samples, the fact that we are seeing such robust changes in small samples might give us some confidence that the brain does change, and that is indeed the basis for growth mindset. So just believing that your brain can change can help students learn, right? And might also change the way that teachers teach. Okay, so on to a new study, and this is a study that we've been working on curiosity. And what's interesting about curiosity is there was a study last year with a single item. They asked parents, how much does your kid like to learn? And that predicted math and reading scores at the end of kindergarten, as well as many other demographic measures, as well as effortful control. And it was especially true for kids from lower socioeconomic status backgrounds. Right? So this makes us think that curiosity is important. There's also sort of a duh moment too, right? That like wanting to learn is going to help you learn. Right? But we wanted to understand what that looks like in the brain. Again, thinking about how these systems get set up in the first place and what that might be for success in the classroom. So we can look to sort of a more classic cognitive neuroscience study of curiosity in adults. And in this study, Gruber and colleagues asked people a bunch of questions, like what does the term dinosaur mean? And they asked them to say, how curious are you about this answer? Right? Some people care a lot. I don't particularly care about this question. I am very glad that my 4-year-old is not interested in dinosaurs. She is interested in rainbow unicorns, which are not actually real, so I'm not sure it's better. But she can tell you all about them. So how curious are you about the answer? And then they said, do these reward regions that Joe was just mentioning track with curiosity? So they looked at the nucleus accumbens, or the ventral striatum, and the ventral tegmental area, the VTA, actually expanded to neighboring dopaminergic areas like the substantia nigra, and they said, how does the bold response track with curiosity? And the answer for both of these areas is positively. When people want to know the answer more, they're showing more ramping up of the reward circuitry. So this is a focused, targeted investigation into these reward regions, but then they zoomed out and they said, where else in the brain is tracking with curiosity? And they found 2 additional areas, an area in the medial wall of prefrontal cortex. This is an area that through meta-analyses we can see is associated with cognitive flexibility and attention, right? That makes sense. If you're curious about something, then you're attending to it more. And actually the strongest cluster was in the cerebellum, which throughout all of my training I was taught pretty thoroughly to ignore, right? Part of the reason is that when you do scanning, you have to say which part of the brain to look at, right? And for most of us, we could only see this part. And this is where what Jeff Aguirre was talking about in the morning really matters. By having better sequences, we can look at the entire brain. I think the era of completely ignoring the cerebellum is over. Actually, studying it has been very zeitgeisty in the past year. So we have these 4 areas. And our lab was particularly interested in this connection here, from the VTA up to MPFC, in part because this part of the brain is very enriched for dopamine receptors. We care about dopamine because it promotes plasticity, right? In many studies in animal models, when you add dopamine to the system, you get greater plasticity. So individual differences in how the VTA innervates parts of cortex is really critical to thinking about individual differences in learning. So when we looked to how to measure curiosity in our sample of 4 to 7-year-olds, we thought about what would be the most ecologically valid way of looking at this. And so we just watched, specifically Ann Park and Ursula Tooley, I'm glad I put this in, these are graduate students who did all of the work, watched as kids interacted with their parents and asked their parents questions. So they read a book without words. It's a very confusing book in which an alien spaceship lands on Earth to encounter a vicious predator, Mr. Wuggles here. They do a puzzle together, and then they explore novel toys together. And what we wanted to know was whether the strength of this VTA and PFC network was related to curiosity defined in this way. So we're looking at connectivity by looking at the correlation of bold responses, as Jeff described this morning. So if 2 areas have more correlation or tighter correlation in bold, they're more likely to be in the same anatomical network. And so here we're looking at the same area that was defined in the adult study, looking at kid questions, and we see this positive correlation here. So kids that have a stronger sort of dopaminergic innervation of these attention areas are more curious. We can zoom out and look at the whole brain. One thing that's nice about this is it allows us just to check that our data track with anatomy. So here we see the cerebellum, right, the whole medial wall of prefrontal cortex. And we zoom out laterally, we see some of these ventral striatum areas and hippocampus, another structure that's critical for learning and memory. These data really track what we know about anatomy. And then we can ask over the whole brain, where does VTA connectivity go with curiosity, as measured by question asking? And we actually see an area that's very, very close to the area that Gruber and colleagues found. Okay. This is a pretty surprising thing to happen, to be honest, in neuroimaging, where you see something so similar to sort of the landmark study in curiosity, and gives us a lot of confidence that we're really looking at the right network here. We see one other area in lateral prefrontal cortex as well. And the last thing we did is we looked at a different measure of curiosity. Parents just saying, how curious are their kids, right? Tracking back to that study in the educational literature predicting math and reading scores. And here we see a different area. We see the cerebellum, which I just told you I was taught over and over again to ignore. And what's interesting about this is that before I ignored it, I just asked a colleague who worked in animal models. I said, what are the connections like between VTA and cerebellum? And he said, funny you should ask that. In Science this year, there was a study with optogenetic manipulation of the connection between the cerebellum and the VTA. And when you turn this connection off, animals don't explore anymore. They don't explore other social animals, right? So from this animal model, we can see that a connection between the VTA and the cerebellum, or more specifically from cerebellum to VTA, is actually necessary for social exploration in animal models, right? So our animal models are giving us biological plausibility for what we're seeing in humans. Okay, so now let's zoom out to the field in general. Okay. So I set us up with these relationships between the classroom, the brain, and the mind. But where I think the field is going is to say that people do things other than reading and math in the classroom, right? There are many lawyers in this audience. I'm sure you have learned a lot about history. Many scientists, too. We care about these things. I think there's a broader focus on the social, cognitive, and affective skills that support learning in the classroom. Curiosity is one of them, But there are other skills that matter, like emotion regulation. If you keep acting out and getting sent to detention, you're not going to learn anything else very well. Right? So I think we actually need to broaden what counts as educational neuroscience if we want to meaningfully understand what leads to educational success. I think another place that neuroscience helps us is thinking about the health of the brain. Right? There are many studies showing major impacts of things like lead and air pollution on learning in school, and neuroscience helps us understand that mechanism. Right? There are things in our environment that set us up for learning across all disciplines and are really meaningful for how education policy should respond to neuroscience. So for example, there's evidence that schools can be sited further away from highways. Right? There's also evidence of changing the school day schedule to accommodate changes in sleep that happen in adolescence. And finally, I think we're going to see better designs that really look at these deltas. Longitudinal designs and even better intervention designs like the ones that I opened with, and connections to animal models. So I sort of, every time I say this, have this funny image of mice sitting in these chairs, right? One might argue that there's not so much we can learn from mice, but I hope that the curiosity finding at least gives you a sense of how the animal models can be helpful, right? When animal models, we can change things causally. We can stress them or not. We can expose them to lead or pollution, things that might be all correlated with something like poverty in humans, and specifically test the causality of these things in a way that I think is more helpful for policy. I think when we start thinking about policy, we have to, again, think about how educational neuroscience is not just a basic science field, but also a translational field. And here are just a handful of issues in my last 3 minutes that I think we'll see more of in the future. So first, I think there are going to be ethical issues around identifying these learning challenges. So a lot of my colleagues advocated for a bill in Massachusetts to do universal screening for phonological awareness for 5-year-olds. Right? That could be a really good thing. You can catch these kids who might have learning issues before they even learn to read. But there might be issues around stigmatization. If you tell a 5-year-old, there's a chance that you might develop dyslexia, and instead of playing during recess, you're going to get early intervention instead, you might start causing self-fulfilling prophecies. Right? Kids just saying, you know what, I'm not going to be good at reading, I don't care. So it's not without challenges. I think that the only way we're really going to learn is to do large-scale RCTs of educational interventions. I would like to see that linked up with neuroscience, but I think we also need to realize that we're going into a very complicated context, it's hard to say, in the classroom. Right? Teachers don't want neuroscience to just show up and change what they're doing. There really needs to be a collaboration between the people who are actually implementing these interventions and the people who want to understand how they work and why. I think we're going to see more and more of neuroimaging in the classroom. There's a really nice study a couple of years ago showing that when teachers lecture, like I'm doing now, there's actually pretty low engagement when you're looking at EEG signals. But when those same students were watching videos and interacting with each other, then you started to see these neuromarkers of engagement picking way up. Right? That might be sort of obvious, but you could imagine ways that teachers could use that information to make their teaching much more engaging. And finally, I think there's a question about whether teachers need to know science, neuroscience specifically. Right? We just heard this issue in law as well. What is the usefulness of neuroscience training? And I think on some level, this is an empirical question. Right? You could do studies where you randomly assign teachers to getting neuroscience training or not. But I think that the quality is really going to matter. So a couple of weeks ago, I gave a talk on curiosity to 800 educators, and I told them some cool things about the brain. And I like to think that was moderately useful, maybe not, but what I think was really useful is that then professors who worked in a graduate school of education made a workshop right after, where they said, okay, this is the psychology of curiosity. This is what we know sparks curiosity. How can we change teaching practices, right, in your classroom to harness kids' curiosity? And I think that's much more effective. So I think we really need to take advantage of the expertise of educators and the people who teach them to bring in neuroscience education as effectively as possible. Thank you.